Hello all, today we are going to discuss the topic that is drug discovery and development. What do you mean by drug? Drug is any substance that is other than food which can be consumed or which can be intake orally for the purpose of treatment or prevention or curing and diagnosis of disease, and the ultimate effect of the drug is which will impact the therapeutic effect, thereby maintain the good quality of health. The drug is made up of two components. One is active pharmaceutical ingredient and the other one is excipients. So what is the difference between the active pharmaceutical ingredient and excipients? Here the active pharmaceutical ingredient, which is the major compound which can source therapeutic activity, whereas excipient is nothing but it is an additional compound that is used to protect the drug and which is used to enhance the stability of the drug. Okay, so that is excipient. So excipient, some of the examples are buffering agents, then preservatives, then coloring agents, etc. And what are the sources of drugs? Generally, the drugs can be derived from various sources such as natural and the other one is synthetic. So in case of natural, some of the examples are plant, then animals, then minerals and microorganisms. In case of microorganism, we can derive the drugs from bacteria, fungi, virus, etc. And the other one is synthetic, and this may be available in the form of either semi-synthetic or synthetic. Sometimes we can go for combinatorial chemistry, so which is the computational method in which we can able to identify the useful components for designing the drug. And the other approach is structure based drug design. So here how the drug will go and bind to the ligand, that we can consider it as structure based drug design. There are three different methods of structure based drug design. One is X-ray crystallography, second one NMR spectroscopy, and the other one is homology modeling. So these are the three different methods in which we can able to predict the drug based on the structure. Coming to the main concept, that is drug discovery. So drug discovery and drug development both are interrelated terms. The drug discovery is the process in which we are going to identify the new potential candidates or new potential medicines, that we can call it as drug discovery. If you want to go for a discovery any drug which may involve various disciplines, that is it is an interdisciplinary field which involves the combination of chemistry, biology and pharmacology. But the drug discovery process will took much more time and it need more money. Okay, and in simple we can say that the process is somewhat quite complex. And why we have to go for a drug discovery? Nowadays peoples are being affected by various diseases, and for that some of the medicines have been identified in order to cure disease. And for some disease still there is an ongoing research going on in order to make the complete cure of that particular disease. So for that purpose we are going for drug discovery. What is the underlying mechanism behind the drug discovery is, so initially we should know about what kind of disease and what are all the genes or proteins that is being involved in that disease, and then we have to go for a screening and identifying and testing new molecular compounds. And the third method is we have to understand both the disease as well as the drug, how they are going to interact with each other in order to produce the therapeutic effect. So that is the underlying mechanism behind the drug discovery. During the drug discovery, some of the most of the drugs may fail. Why? There are two reasons. One is it may not work effectively, and the other one is it may produce toxic effect instead of producing these instead of producing the therapeutic effect. So For that reason, the drugs are being failed. So this is the schematic diagram represents the drug discovery and development process from the target identification till the compound is being marketed. That is the diagrammatic representation. And during the drug discovery and development, the drug may undergo different applications such as investigational new drug application and new drug application, that is NDA, or biologics license application, and food and drug administration. So these are the different regulatory authority if you want to release the drug into the market. So the drug has to undergo these different regulatory authorities, and it may reach the patient. And what is the function of these regulatory bodies? So this regulatory body, which may act as an interface between the drug discovery team and the regulatory authority. So through that we can able to communicate whether the drug is being working properly or not. So in order to identify the pros and cons of the drug regulation, and in what way we can able to improve the efficacy of the drug with the help of this drug regulation. And what is the role is they can communicate with the health authorities, and they can review and approve the drug, and whatever the documents which they are going for reviewing and approving, all the documents has to be maintained, starting from the internal audit till the registration. And we have to keep on update the submission and filing of drug application. So for that, this regulatory authority play an important role in giving the valuable inputs to the drug discovery team in order to make the drug in much more effective manner. And this is the typical diagram represents the stages of drug development. So the drug development starts from drug discovery, then it may undergo preclinical development, and the third one, that is clinical development, and fourth one, that is approval, and last one, that is market. Under the drug discovery, initially we can start with more than 10,000 compounds, and from the 10,000 compound only one compound is going to be approved, and that compound is will be available in the market. Okay, so drug discovery involves various steps such as target identification and validation, and the second one, that is lead discovery, and third one, that is lead optimization, and last one, that is drug development. So this figure represents the drug discovery, that is the different stages in how the drug is being discovered. So first one, that is target identification. So this is the preliminary step in the drug discovery. Here you should know about what is target. Target is what we can say is which may be of any macromolecule, which may be a protein or a genes or RNA that is being associated with the disease. Okay, so this target should be druggable. Druggable is nothing but the when we are adding the drug to target that particular gene, so the drug will go on interact with the gene, thereby which may produce the therapeutic effect, that is that is druggable property. So there are several tools have been have been used for the target identification. Which is being given on the uh, right hand side of the slide. Okay, so if you want to identify the target, we should know, we should have a basic understanding about the various principles of cell biology, molecular biology, biochemistry, then genomics, proteomics, and bioinformatics. And various methods which is used to identify the target is by means of data mining, and with the help of biochemical pathway we can able to screen the particular enzymes and receptors and genetic association. Through that we can make interrelation between the disease and drug target compound. And the next one is phenotypic screening. So here we are going to identify the targets. And the last one, that is expression profiling, so in which how the mRNA and protein level is being expressed in that particular disease. And coming to the next one, that is target validation. So once we have identified the target should undergo the validation process. So this validation starts from in vitro methods and in vitro methods. Nowadays, there are various tools and techniques are being used to identify the disease. And some of the methods that is being used for target identification and validation is given, such as gene expression, biochemical assay, and specifically in the biochemical assay, cell-based bioassay is being commonly used. And we can use in silico methods, then proteomics, genomics, and imaging and analysis. As I told that druggable is that drug will go and bind to the target, thereby which may generate and response, that is druggable. And the next step after the target identification and target validation, the next one is lead discovery. So lead discovery is what we can say is, initially we start with 10,000 compounds, and from the 10,000 compounds we can select some thousands or hundred of compounds, and out of those hundred we have to select which one is binding very effectively to the target, and how it is being producing response, that is pharmacokinetic mechanism or pharmacokinetic properties. So based on that we have to identify the lead compounds. Okay, and here, so whatever the compound that is being identified as a lead, this will be enter into the preclinical stage. And once it is being entered into preclinical stage, it may undergo various animal study, and it may enter into the clinical, and it may be considered as a potential candidate drug, and that drug may be marketed. Okay, so that is lead discovery. So here we are screening the drug compounds out of 10,000. Okay, so here we can say that it is a hit molecule. So the definition of the hit molecule may be vary according to the researchers. And in general, hit is nothing but the one which produce the desired activity in a compound screening. And the activity have been tested again and again, whether the compound is producing the same response or not. Okay, so there are different methods to identify the hit molecules. One is high throughput, then focus screen, fragment screen, virtual screen, physiological, then NMR. So here what we will do is, we can take the drug compound, and we may make those compound in order to test whether it is being working at a cellular level, molecular level, and biochemical level. That is lead discovery. And we have to consider several factors during the screening assay. One is reproducibility of the assay, then pharmacological relevance of assay, interaction of the compounds in the assay, then cost and quality. Reproducibility, whatever the result we expect in the initial stage, and the same result is being coming out in repeated manner or not, that we have to check. And pharmacological, whether the drug is being produced only the therapeutic effect, and it doesn't go and bind to any other non-target cells instead of binding to the target cells. And the interaction, we have to keep on monitor how it is being effectively binding. And cost, the lead discovery compound with involves a high more money, and the quality we have to produce the best quality of the drug so that it may produce the very good therapeutic response, so in which all the patients can able to afford it for the treatment purpose. And one rule, that is Lipinski rule. This Lipinski rule, it is otherwise called as rule of five or Pfizer's rule. So, which was formulated by Christopher A. Lipinski in the year 1997. What he found that since most of the drugs are being administered in orally, and the oral form of drug delivery will be very small, and all the oral administered drugs are available in lipophilic compounds. So whether it is being producing the particular pharmacological or biological activity in response to the physicochemical properties of our body. So that rule was framed by Christopher A. Lipinski. And this Lipinski rule helps to distinguish drug-like and non-drug-like molecules, as well as it may predict high probability of success or failure due to drug-likeness for molecules complying with two or more of the following rules. So here is the Lipinski rules. So the rules are as follows. So one, the molecular mass should be less than 500 Dalton, and the second one, that is high lipophilicity, and third one, that is the less than 5 hydrogen bond donor and less than 10 hydrogen acceptor, and the molar refractivity should be between 40 to 130. So once the drug discovery team have identified the hit from the library, and then they have to go on whether the compound is being working efficiently or not. So what they have to do is initially they have to remove any compounds that are known to frequently show up in screening test but may not be as useful for the specific research. Next, they may go for combinatorial chemistry, in which various combinatorial algorithms or computer programs have been used to consider whether the drug has a good response or not. And then we have to go for optimizing the dose. Okay. So these are the various assays that can be used in screening. So such as aqueous solubility, then log P 7.4, then microsomal stability, CYP 450 inhibition, etc. And the respective what why we have to go for this particular assay. So the respective function of that assay is being given in the comments, that is in the table. And lead optimization, so once we have identified the drug, that drug has to uh, bind to the target. For example, if you have a screen drug which, in which the structure is somewhat different, so we have to optimize the structure, as well as we have to improve the efficiency. So for that we are going for this lead optimization method. Okay. So at this stage, what the researchers have to do is, so initially, so in the previous step, that is lead discovery, so they have to go for synthesizing the hit that lead molecules. In if the compound fails during the preclinical and clinical stage, so for that they have to go for synthesizing the same compound. Okay, so we have to provide the favorable conditions in order to generate therapeutic response, such as pharmacokinetics and pharmacodynamics, and uh, absorption, distribution, metabolism, and excretion. Okay, so what we have to do during this lead optimization, we are going to optimize the structure and we are going to improve the efficacy of the drug. And there are various tests in which the lead optimized compound will go. One is genotoxicity as testing, then general behavior testing, then high dose pharmacology, then pharmacokinetics and pharmacodynamics, then dose linearity, then repeat dosing pharmacokinetics, metabolic profiling, then chemical stability and salt selection. So why we have to go for these tests in order to validate whether the molecule is being working functioning properly or not. Okay, then only we can go for the trials. So if we want to produce the lead molecule, it may took much more time. And preclinical research, so in which the clinical trial will be happened in case of animals before it is being tested in humans. That is a preclinical development, in which we are going to evaluate the safety, efficacy and pharmacological properties of the drug candidate. Okay, so whatever the data which we are collecting or recording during this research, it has to be maintained so that the further evaluation can be carried out before the drug can be administered into humans. So these are the different steps involved in preclinical development. One is safety evaluation. We have to check whether the drug is being producing the therapeutic effect, and then pharmacokinetics and pharmacodynamics, the drug has to work with the body effectively. Then optimization of dose, we have to start with a little amount of dose. Then efficacy testing, then ADME process, then formulation. So during the formulation we start with less quantity, then toxicology studies, then reproductive and developmental studies and clinical development. So after the drug has been tested in the animal, it may be tested for the human. Okay, so in which we are in need of several human participants, whether the drug is being working properly or not. So here we have to go for different phases such as phase 1, phase 2, phase 3. And the difference between the phase 1, phase 2 and phase 3 is the number of volunteers who are involving in the trial. That is the main difference. And coming to the phase 1, here the healthy volunteers, we have to check the safety, metabolism and dosage. And phase 2, here the volunteer quantity will be somewhat large, and that can be done with the patients. And third one, we have to go for a large group of participants to confirm whether any side effect is being there, or the treatment method whatever which is being available, we have to compare. Okay, and some other phases such as randomized control trials. So here the trials with random assignment to different groups for unbiased, that is whether the result is coming properly or not. We should not expect unexpected results. Okay, and informed consent. So before making the humans as volunteers in the clinical development, we have to inform, so this is the drug and we are going to administer into you. And sometimes it may we will inform the pros and cons, what will be the risk and what will be the benefits. Okay, and then we have to go for monitoring and safety, and we have to interpret the data, and we have to go for regulatory approval. So out of 10,000 compounds, only 1 in 10 candidates reaching the market, but at this stage the financial consequences of failure are much higher. Regulatory approval, once the drug has been successfully undergo the clinical trial, the regulatory bodies such as FDA, EMA, etc., they may analyze the data, and which may involve review, then safety monitoring and risk benefit analysis. Once they gave approval, the drug can be marketed and which can be prescribed for specific medical conditions. Okay, and the approval process will vary from country to country. Okay, and these are the different steps which is involved in the approval of drugs by the regulatory authorities. So initially we have to get the application form, then we have to submit it to the concerned regulatory authorities. Then the regulatory authority will review whether the data whatever which is being given is correct or not, and they may go for evaluation. And there may be some advisory committee which may act as an independent evaluation, and they go for, then we have to monitor the drug. Then labeling and packaging, so this labeling and packaging will be also monitored by the regulatory authority, whether the dosage or the name of the drug, then manufacture, expiry date, whatever is given correct or not, they will check. Then the risk benefit analysis and post marketing surveillance, then conditional approvals and fast approval. For example, if any of the drug is being producing any side effects, so we have to go for this fast track program, and we have to um, stop the production. Then finally the approve the decision which is given by the regulatory authority is final one. And marketing of drugs and post marketing monitoring, once the regulatory body approve the drug, then the drug may come into sales for the pharmaceutical company and the healthcare providers, so that the drug will be distributed to the consumers and the patients. Okay, so here the production, distribution and sale of pharmaceutical products to meet the medical needs and improve patient health. Okay, so once it is for example, if a drug is being produced in India, then it may reach to different parts of the world, so that on those who are suffering from that particular disease can get benefit. Okay, so that is marketing and post market monitoring. Thank you.